So enzyme kinetics. So like I mentioned before, there are different ways to measure the reactions catalyzed by enzymes. The first one is the Michaelis Menten. So in this plot, the enzyme concentration is constant, but the substrate concentration is increased. So so you will have a given concentration of enzymes that doesn't change, but the only thing changing is the substrate concentration. So there are a few parameters you need to know. The first one is Vmax. So that's the maximum velocity of the reaction with the given enzyme concentration in the experiment, or the reaction with the enzyme. And Vmax can also be measured as Vmax equals E times Kcat. So E is the enzyme concentration, and Kcat is what we call the turnover rate for a single enzyme. So that's pretty much the rate of catalyzing the enzyme, catalyzing the reaction by a single enzyme. So that makes sense, since if you take the rate of a single enzyme and multiply it by how much enzyme you have, you'll have the maximum velocity that you can, maximum velocity of the enzyme with the given amount of enzyme concentration. And the next parameter is called Km. So this is the substrate concentration when the velocity of the reaction is half of Vmax. So the velocity is occurring at half its maximum velocity. For Km, you need to know that if a reaction has a high Km, that means it has a low affinity. The enzyme has a low affinity for the substrate, which makes sense, since if you need more substrate concentration in order for the enzyme to bind to the substrate and catalyze the reaction, then the enzyme doesn't really have a high affinity for the substrate. While if you have a low Km, that means you only need a small amount of substrate in order for the enzyme to be working at half its maximum velocity. So only a small amount of substrate is needed to reach half of Vmax. So this is a standard Michaelis Menten plot. On the bottom, the x-axis is the substrate concentration. So this is increasing as time goes by. And then on the y-axis is negative 1/2 Vmax. So this is the measuring the velocity. Oh no, sorry. The y-axis is the reaction velocity, V. And on the bottom you can see Km. So this is the maximum. This is the substrate concentration required to reach half of Vmax. So you can see it's 1/2 of Vmax on the y-axis. So at half of Vmax, this is the amount of substrate concentration needed. And you can see as Km goes more to the right, more substrate concentration is needed to reach half of Vmax. So the enzyme will have lower affinity for the substrate, while if the Km is to the right of this Km on the chart, less substrate concentration is needed to reach half of Vmax. So it has a higher affinity for the substrate. Sorry, you said, oh, well, you said repeat that. Which part do you mean? Affinity and concentration needed.
Yeah, you said to the right you would need more, and to the left we need less of what?
So to the right of this Km, if Km is to the right, you see on the bottom it says substrate concentration. So if Km is moved more to the right, you'll need a higher substrate concentration. So that higher substrate concentration needed to reach half of Vmax, that means the enzyme will have a lower affinity for the substrate, because you need a lot more substrate in order for the enzyme to bind to the substrate and catalyze the reaction. And then similarly to the left, if Km is moved more to the left on the x-axis, it requires less substrate concentration. So less substrate is needed for the enzyme to bind to the substrate and reach half of Vmax. So that would signify a higher affinity of the enzyme for the substrate in question. Okay. And so the next one is called the Lineweaver-Burk plot. So it graphs the same data as the Michaelis Menten, but it's graphed differently. So as you can see, the Lineweaver-Burk plot, it has a standard slope, has a constant slope, so it has a standard line as that's plotted, while the Michaelis Menten has more of a curved graph. And yeah, so the Lineweaver-Burk, it just tries to simplify the data that is graphed as a Michaelis Menten plot. It uses the reciprocal of the Michaelis Menten plot axis. So as you can see here, the Michaelis Menten, it plots V, the velocity of the reaction, as the y-axis, and substrate concentration S as the x-axis. While on the Lineweaver-Burk, it plots the reciprocal. So it plots 1 over the velocity of the reaction as the y-axis, while the reciprocal of the substrate concentration, so 1 over S, is the x-axis. And so as you can see on this graph, it's kind of hard to identify 1/2 Vmax and Km, because you have to guesstimate where the middle is, where the 1/2 of Vmax is, and then you have to guesstimate where the substrate concentration is. Lineweaver-Burk plot simplifies it. So it shows that the x-axis is -1 over Km. So you can use the x-axis intercept to identify the Km, so the substrate concentration, to reach half of Vmax. And then y-axis is 1 over Vmax. So you can use the y-axis intercept to calculate the maximum velocity of the reaction. So Lineweaver-Burk plot and Michaelis Menten, they're pretty similar. It's just how they're plotted. The Michaelis Menten is good for looking at the overall velocity of the reaction, but it's hard to determine the 1/2 Vmax and the half Vmax and Km. So Lineweaver-Burk plot just simplifies it by allowing you to calculate it using the x and y axis intercepts.